Lau from King's College.
Kayla Lee from PT College.
And I'm Sita from West Island School. And here's our research title, Behavioral Difference of C. elegans when they encounter different bacteria.
The reason why we choose C. elegans as our test subject is C. elegans are transparent nematodes that can be found in soil, and in laboratory, they mostly feed on E. coli for survival. But by conducting this research, we would like to find out if there are any other bacteria that attract C. elegans or repel them.
First, we isolated and characterized bacteria from soil samples. Second, we allowed C. elegans to grow in the presence of E. coli and the newly isolated bacteria. Finally, we analyzed the food preference of C. elegans by scoring the number of worms found in different areas of the test arena.
Methods. To obtain bacteria, we collected soil samples from the countryside near HKUST, and we collected soil samples from here. And bacteria were retrieved from soil samples by plating on an LB agar plate, and we did 6 colonies per plate per student. And the isolated colonies were incubated overnight at different temperature, so at room temperature and at 37 degrees Celsius. And also we test for their resistance to antibiotic ampicillin by placing them with ampicillin at room temperature. And to identify the bacterial species, a segment of their 16S ribosomal RNA gene were sequenced. We did it by doing the polymerase chain reaction to amplify the target fragment. Then we use the correct PCR products verified by size to send for sequencing.
And after we do that, we sequence the DNA of the bacteria and identify the species they belong to. And this is the graph showing the sequencing results of the bacterial DNA. And we examine and record the preference of C. elegans to free sequenced bacteria against E. coli strain that is used as the standard laboratory diet.
This is a result showing the conditions that favors the growth of isolated bacteria. And here are the photographs of different bacteria on the agar plate. And here is a chart showing their preference. And the tick represents growth. And Z1, Z3, and M2 bacteria were chosen to further proceed in the experiment.
Sequencing results. Attempts were made to amplify fragments of the 16S ribosome DNA gene of bacteria Z123, M1, M2, H1, 3, and 4. Purified PCR products were sequenced. And this is the chart where the bacterial species and strain.
And food preference assay. Worms were allowed to grow in the presence of E. coli and test bacteria from L1 stage, and the food preference assay was scored when worms were at L3, L4, or 1 day old adult stage. And this is the formula for calculating the food preference index. And the image on the right shows the placement of test bacteria and E. coli in 2 different regions.
And this is the chart of the result of food preference index.
1 day old adult C. elegans prefer Z1 to E. coli, whereas L3, L4 larvae C. elegans prefer E. coli to Z1.
1 day old adult C. elegans prefer E. coli to Z3, whereas L3, L4 larvae C. elegans prefer Z3 to E. coli.
L3, L4 larvae C. elegans prefer M3 to E. coli. And we found that C. elegans at different stages of life, 1 day old versus L3, L4 larvae, seem to have different preferences for bacteria. That's the end of our presentation.